start, and I missed the presentation of our speaker. So today we have Gaspar Jackley. He's from Life Science Institute here, very close to us, and he's kindly accepted to be talking to us. So if you are here, don't be afraid, you're in the right. This is a CS seminar, even though the title sounds a little bit far away from us, but this is also something that is important for us as computer scientists, because usually we are not working at the end, like delivering things, but most of the time we are just the means to provide technology or tools for others. So it's important for us to bridge this gap between multidisciplines. So as I said, Gaspar kindly accepted us our inviting. He has a PhD in biology from 1999 in Budapest. He was a postdoc at EMBL in Heidelberg. He was a group leader for 10 years in the Max Planck Institute for Developmental Biology. And he joined LSI, as I said, as a professor of neuroscience in 2017. So his research interests mainly focus on structure, functions, and evolution of neural circuits in marine ciliate larvae. And he also wants to study the origins and early evolution of neural systems. So without any further, Gaspar, the floor is yours. Thank you.
Thank you very much, Diogo, for the introduction and invitation. It's a pleasure to be here. So I will give an introduction into this animal model for circuit neuroscience. And I will start by a general introduction, what we are interested in. So we are interested in understanding animal behavior and more broadly cognition in the context of entire animal bodies. So not just parts of the brain, but how the brain comes together with the rest of the body. And there are two kind of important concepts here that are also known from areas like robotics, computer science. One is embodied cognition, which means that actually the entire body of the organism is central for perception and cognition and action, and through these reafferent relations, which mean that as this body or animal moves, all its sensory perception will be influenced by its own movement. And this is called reafference. So any effect on an organism's sensory mechanism that is due to the organism's own action. Just think about when you are moving, then the visual field is moving in front of your eye. And this is very important information that the nervous system also has to process. So how does it come together in full bodies? And of course, neuronal circuits are very important. It's one of our key interests. And in the nervous system, we know that neurons are linked into intricate networks by synapses. And this can nowadays be mapped by highly powerful electron microscopy techniques, where people can reconstruct nervous tissue. And this is a very spectacular example from a part of the human brain. And the problem is that even with the most powerful and expensive technology nowadays, it's still only a very tiny fraction of the mammalian brain that can be reconstructed at synaptic resolution. So to really get to what is called the connectome. So how do the neurons connect to each other? And with respect to the entire mouse, this is just still a tiny fraction. So it is hard to understand then these reafference or embodied relationships in such a large organism at the level of individual neurons and circuits. And this is one of the reasons why we are studying a much smaller organism. And this is this marine annelid that is called Platynereis. And these are some pictures of the early developmental stages of this animal. This is a marine worm that can be cultured in the lab in the LSI basement, or we have a big animal facility where we keep these animals in seawater and we feed them and can breed them. This is the adult. And then we can obtain these larvae every day of the year, and we can do experiments with them to study their nervous system and behavior. This is how mating works. These are two adults, a male and a female. They perform this nuptial dance, and they release the gametes and the eggs and sperm. And then after a few days of development, we have hundreds of these synchronously developing larvae that are swimming that we can study. And in nature, these larvae would be part of the zooplankton, so they would be swim freely in the ocean. And we are interested in how they react to various stimuli, for example. And in this case, this is light, directional light stimulation, and the larvae will swim towards a light source. This is called phototaxis. This is very important and widespread behavior, and one paradigm where we can really start to dissect these reference relationships and this embodied cognition, so we can try to understand this in a whole body context. So how does this work? This is the larval stage that performs this phototactic behavior. And they have these hairs here are cilia. So these are these cellular structures that beat, as you can see in this video. This is the top view. And as the cilia beat, they propel the larva in the water. So this is really a locomotion pattern. It's not based on muscles like when we move, but based on these tiny cellular protrusions, the cilia. And as the larva moves, it uses its very simple eyes to scan the direction of light. And these are really the simplest eyes in nature. You can also call them proto-eyes. So these only consist of two cells, one photoreceptor cell shown here, and the pigment cell, which shades the photoreceptor cell from one direction. So you can imagine that this photoreceptor will only receive light from one side. And this anatomy is the basis of the directional sensitivity of these larvae. So this is how the 3D reconstruction of this eye looks like. And if we reconstruct by serial electron microscopy the anatomy of this eye photoreceptor, so the rhabdom is the red territories where light is sensed. This is the cell's body, and there's an axon, and then it forms synapses directly on the cells of this ciliary band. So this is the motor of the larva. So this suggests that there is a direct sensory motor connection in the system. And we could show that when we stimulate this eye by light in the microscope, then there is a change in the beating pattern of the adjacent cilia. And the water flow slows down. This is just the bead that is tracked. And if we illuminate the eye, then the flow slows down on the illuminated side of the larva. And this is the basic mechanism of steering. And this is reminiscent of this idea of Valentino Breitenberg, these vehicles, which are just conceptual kind of mini robots able to turn towards the light by just this very simple wiring, and these wires and sensors regulating the speed of these wheels. And so we tried to understand in more detail this locomotion pattern, and this was a computational project in collaboration with François Nedelec, who built a computer model of this larva, which was modeled as a sphere. And these arrows here represent the forces generated by the cilia that propel the larva, and these colorful blobs here are the eyes with a small arrow that shows the direction of the sensitivity of the larva. And then we can model the movement of the body and how light will influence the translation and the rotation of this body and simulate trajectories of swimming. And we can get beautiful phototaxis with this model in the computer and then analyze all the parameter space. There are two very important parameters in the model. And I will just talk about the spin parameter, which essentially describes how much the larva is rotating along its anterior posterior axis. And it turns out if we switch off this body rotation, then there is no phototaxis. So we score here the angle of trajectories towards the light. And if you don't have spinning, there is no phototaxis. You need to have some degree of spinning, either left-handed or right-handed, to be phototactic. And the thing is that the Breitenberg vehicle, right, it only moves in 2 dimensions, it's not swimming at all. So actually that mechanism is unlikely to work in nature. So if you look at a real life larva, it's indeed spiraling along its anterior posterior axis. So these are really helical trajectories with these parameters. And you really need to put in this helical rotation into the model to get the direction of swimming. And the reason for that is that as the larva is moving in this helical fashion, you remember these directional eyes. These are moving and scanning the visual field, just like a radar is scanning for objects by this rotation. It's a similar principle. And what it detects is fluctuations in the light intensity. So you can imagine a rotation of a sensor, and there's a directional light source. Then you will get this sinusoid modulation of the signal as the sensor rotates. And this is what then translates periodically to a steering effect on the on the ciliated cells. And that's how you get phototaxis. So what is the lesson from this? The lesson is that this is really the simplest behavior you can imagine. There are a lot more complex behaviors, walking, eating, mating, whatever. This is really just swimming towards the light. There's very few behaviors that could be simpler than this. But here also already we could show that even though we know the entire connectome, we know that this cell connects through synapses to that cell, we could only understand the behavior when we placed it into the context of the whole body. And we have to understand the body rotation and how the sensor is scanning the visual space to understand even this simpler behavior at all. So we really need whole body approach for this, even though it's kind of seems like a simple reflex. It's actually a lot more than that, because this embodied sensing and reafference are essential components in this behavior already. So what about other behaviors? There are many other types of behaviors that we can study in this organism, including crawling and startle when they are frightened, or how they interact with the predator, like this jellyfish. And we are interested also to study and understand some of these behaviors at the level of circuits and the level of the whole body. And to really do that systematically, what we wanted to get is the connectome of this entire animal. So not just a part of the brain, but really get all the neurons and all the connections. And it's small enough so we can size it up in the electron microscope and then image all the sections at nanometer resolution, and then reconstruct all the neurons and all the other cells and find all the synapses. So what we have is roughly 2,000 neurons in this body, 30,000 synapses, and over 1,000 so-called effector cells. So these are like muscles and the ciliated cells, and it's a body of roughly 9,000 cells. And of course, this is what electron microscopic images look like. So we can really look at the fine ultrastructural detail of all the cells. We can recognize muscle cells. We can recognize cells that have some skeletal elements and so on. And we can recognize the synapses, and then build up basically this cellular model of the entire body. So these are just the different types of neuronal cells. There's an incredibly high diversity of types of neurons. There are over 180 different types of neurons. And type here means really cell type based on the ultrastructure, morphology, and connectivity of these cells. There are 50 different muscle cells and many other types of cells. So this is kind of a big data approach, no? And of course, this kind of connectomics is more and more done by automatic segmentation and analysis of these large multi-terabyte image data sets. So this is really a high challenge for computer science, and also the underlying analysis, not only the image segmentation and merging and splitting, but also then later the analysis of the connectivity. So these are examples of synapses. This cell connects to this cell through a synapse. And if you have all the cells and all the synapses reconstructed, what you can get out of this is a large network, which we call the synaptic connectome. So every node here, it represents a cell, a neuron or an effector cell, such as muscle or cilia. And every connection is basically synaptic contact. So it's a directed graph. So cell A synapses onto cell B. There are arrows on it which you don't see. And then you can use all the tools, of course, of graph theory to analyze these kinds of networks. And there will be more and more of these networks coming, increasingly larger. So this is maybe 2,500 nodes, but as electron microscopic projects progress, there's now a network for the Drosophila hemibrain, which is, I don't even know, maybe 40,000 cells, so like giant numbers. And there's a lot to be done and understood and analyzed with the tools of computer science here. What we did here is, for example, subdivide this network into various modules based on community analysis, and then we find various anatomically and functionally distinct modules that we can then go into and understand in more detail how those work. Now, I would like to highlight another network that we can get from this data, and this is basically the network of the entire muscle system of the animal. And what you see here is a muscle connecting through some skeletal cell. It's a cell that has this so-called acicula. It's a chitin rod. It is like our bones. And there are these connections between these. These are adhesive connections. So this is basically force transmission. And these are called desmosomes. This is analogous to, for example, in our body, a muscle connecting to a bone through a tendon. And when you contract the muscle, this is where the cell can pull this other cell. So there are over 800 muscle cells in the body, and we found all the desmosomal partners for all these cells. And this is how it looks like over the body. So these 800 muscle cells, which are shown in this orange color, connect to over 1,000 of other cells through these force transmitting desmosomes. And what you can get from this is the so-called desmosomal connectome. So it's another connectome coming from the same body. These are, in this case, not synapses, but these are the desmosomes that connect the different muscles to many types of other cells onto which they pull, skeletal cells and epithelial cells and so on. Now, this is an undirected graph, no, because these desmosomes don't have a direct direction. They basically link two cells. And this graph is, in a way, a representation of another aspect of this body. And what we are really interested in is try to see how the neuronal connectome graph connects to this desmosomal force connectome graph of the muscle system. And I highlight this in one example. This is what we call the postural control system, and this system controls how much the animal bends its tail to the left or to the right. And this is happening through these red motor neurons that are innervating these long muscles along the trunk of the body. And when these muscles contract, then the larva bends. And we can represent it like this. So you have to imagine this as kind of two overlaying connectomes. We have the desmosomal connectome, which is the muscle system plus force transmission. And then to this, there are synaptic connections coming from the motor neurons, because the motor neurons connect through synapses to the muscles. And these 4 motor neurons, for example, connect to these red cells here, these longitudinal muscles. And when these muscles contract, these connect through the desmosomes to parts of the body. And of course, the muscles don't just connect in themselves. When they contract, they have to pull something. Like in our hand, it's the bone that is pulled, so you can move your arm. And it's the same principle here. These muscles connect and pull on these other cells so that the larva can bend. So what from a computer science perspective, I think is very interesting, and I think requires a lot more work and potential area of collaboration is these multi-layer networks. And these are just 2 layers, but there are many other layers, for example, chemical communication between these cells, and how to analyze these, for example, when you overlay a non-directed graph on a directed graph, how to analyze even the principles of these graphs, how they are built and interact. So what you see here is exactly this postural control system being activated, and you see the bending of the body. This is when these longitudinal muscles in the trunk contract, and in this case, the larva is turning away from a light stimulus. So these are eyes that develop later than the eyes I talked about earlier. These connect to these big red motor neurons and turn the trunk through this network, which I'm not going to show the details, but basically this is also a behavior when the larva turns in a changing to a changing directional stimulus, light stimulus. Okay. As this is something I'm going to skip. So I'm coming to the next example where we can nicely illustrate that a full body approach is essential to understand behaviors and how the nervous system coordinates the entire body. And this is the example of the coordination of the cilia. So this is a later stage larva, and this has more bands of the cilia that they use for swimming. And what is fascinating about this, even though these are distinct cells and distinct segments of the larva, the cilia show an activity pattern that is coordinated. So you can see that the different bands close and start beating at the same time. These are chymographs showing the activity over time of the cilia. You can see that here is a closure when all the bands stop, and then here they resume. And again, you see that it's synchronous across the whole body. So how does the head know what the tail cilia are doing? How is it coordinated? And to understand that, we did all kinds of neuroscience analysis, including analysis of neuronal activity by looking at these calcium indicators that reveal the activity patterns of neurons. And we found neurons that show an activity that very strongly correlates with the activity of the ciliated cells that bear the cilia. And whenever these neurons are active, then the cilia shut down. So, and these neurons, some of them remarkably actually are very big and innervate all the cilia in one of these bands. So this is one example. It's a head motor neuron that innervates all these 23 ciliated cells in the head in the most anterior ciliary band. And when this cell is active, it shuts down the cilia. And we can also show some more experiments. If we kill this cell by a laser, then we don't see any more these regular closures. Okay, and in the trunk of the body, there are corresponding motor neurons that again span the entire body and innervate all the segmentally arranged ciliary bands. And when these cells are active, they synchronously then shut down all the cilia of the body. So this is a circuit that spans the entire body. It's rhythmically active. It's driven by a pacemaker circuit. And whenever these neurons are active, then in a coordinated manner, all the cilia stop beating. And these are just some examples of these beautiful neurons reconstructed by the EM and labeled by a transgene. So the key point here is that, this system, I'm gonna skip this, also spans the entire body and is driven by a pacemaker. And also we need to understand the entire configuration of this. Do you see my share?
No, we can see. Like we can see the slide deck, but was not.
Yeah. Okay. Now it should be fine. Yeah. Okay. So again, a whole body approach was essential to understand how the system works. And this is the third example where it's again a relatively simple reflex, but again, it's a whole body response. And what you see here is that we are startling the larvae by just banging on the microscope stage, and there are vibrations in the water, and the larvae sense that and stop swimming and throw in the air, so to speak, their legs, these parapodia, which have these chitin bristles. They are also called bristle worms because they have these bristles. So how does this work? We could show that this response is triggered by near-field hydrodynamic disturbances, so you don't have to touch the larva. It's sufficient to vibrate this tungsten needle from a certain distance from the larva, and then you can trigger this response. So it's really hydrodynamic vibrations that are sensed. And you see this white signal. These are tracer beads in the water. So you see the flow and you see how the flow stops. And this happens when all the cilia stop in the body. And again, this has to be coordinated because it doesn't make sense to just stop some of the cilia. If you really want to startle and stop swimming, you have to stop all your motors across the entire body. How does this work? What are the sensors here? This is a very different type of sensor than the eyes I showed before. These are kind of small antennae, which are called mechanosensory receptors. And this is a cellular protrusion. It's again a cilium, but it's not non-motile. It's a sensory cilium, this green cilia shown here. And these can pick up like an antenna these hydrodynamic vibrations and transfer those to neuronal signals, which we can show in this experiment. This is a larva. Here you see two of these mechanosensory cells, and we stimulate them with the tungsten needle, and then we measure the activity of the neuronal cell bodies, which are deep down here. And you can see that whenever we provide the signal, these so-called CR or collar receptor cells respond to these vibrations. So this is how we can detect and know which cells are responding to a certain type of stimulation. And then once we identify the cells, we can go to the electron microscopy data set and find the same cells based on their position and specific ultrastructure, and then go to the connectome of these cells. So this is the position of all of these vibration-sensitive neurons in the whole body. And these are then connecting through synapses to these blue interneurons and these other cyan interneurons. So what you see here, just bunch of neurons, but eventually the circuit innervates all these muscles, and these muscles are responsible for contracting the body and lifting the legs of the larva. So this is a summary diagram of this neuronal network. So what you see here, every node here is now a group of cells. So there are 25 of these vibration sensors in the head and 10 of them in the tail of the larva. And again, these arrows are synaptic connections. So these cells talk to each other through this network of interaction. There are various interneurons, and the red ones are motor neurons. And there are two kind of pathways in this circuit. One goes to the ciliary cells, and this is the same, these red ones are the same ciliomotor neurons that shut down the cilia. So basically this system engages the same control system for cilia to stop the beating of the cilia. And then there is this muscle motor branch that goes to 55 different muscle motor neurons that then innervate all these muscles that will lead to the shortening of the body and the lifting of the parapodia. And this is also a quite complicated network, but it allows the whole body coordination of this action. And this is, for example, ensured through intersegmental connections. So remember that this trunk has 3 segments, all of them with their own legs, which look very similar. And how is it possible that if you have a signal on the head, you can actually reach all 3 segments and trigger a response in all the segments? This is ensured because the head sensory neurons connect to also deeper in the segments to interneurons, and those then connect to motor neurons across all 3 segments. So both stimulation to the head and the tail through this interconnected network can ensure actually the activation of all the muscles in the entire trunk. So it's not just one segment will contract, but all the segments because of this intersegmental coordination. Now, I skip this. I wanted to show that we also have genetics in this system, so we can look at specific genes involved in some of the behaviors and then mutate them. This is a mutant that does not show this mechanical sensitivity. It's an ion channel, a specific ion channel, also related to some human ion channels involved in some diseases. And these ion channel is specifically expressed in these mechanosensory antennae cells. And if we, by CRISPR, knock out this gene, this disrupt the gene, then you see that these larvae don't respond anymore to the stimulation. And then we can play with these mutants, and the startle responses now is in nature, it's generally considered to be important to avoid and detect predators, which come and eat you, and you want to startle and extend your bristles to kind of save your life. And these Platynereis larvae in the plankton have these predators, these very scary looking so-called copepods. This is a crustacean. And so we can also get these in the lab. And you can see we can do this predation assays. If this copepod attacks the Platynereis, you see that it throws up its legs and can escape. This is so-called wild type, so it's not a mutated. And here is the mutated larva that lacks this ion channel. And this one cannot defend itself, so the copepod will eat it. And there's a high difference between the wild type and mutants. So the mutants have a much larger predation rate, so they are more exposed to this predation. So we can also link kind of these behaviors through genetics to a real life situation or an ecologically relevant situation. So the way this works, you have to imagine that the Platynereis is, as it's swimming with its cilia, it is creating a flow, because every swimming organism will have kind of a specific signature in the water, hydrodynamic trail. And these copepods, what they do, they have these very long antennae, which are mechanosensory, and they can follow these trails, and that's how they track their prey. So it's a hydrodynamic predator. And of course, if the larva detects that there is a predator nearby, it makes sense for this larva to stop generating this cue, to stop beating with the cilia, because that's how you are detected. It's like the cycads stop chirping when you go closer to them. It's a similar principle. So they have to shut up because this swimming is the cue that tells the predator where they are. That's why it's a very important response. And once the predator found you, the only thing you can do is struggle with your bristles. So is this behavior simple, and are these worms simple? I just want to remind you that this is a worm, the bobbit worm. They can eat entire fish. So it's not always the vertebrates that are the so-called higher organism. Of course, there are no higher and lower organism. In this case, it's obvious that the bobbit worm is a much higher organism than the fish because it eats the fish. And of course, we cannot think about these also relatively simple organisms as simple or just having simple reflexes. And I argued that some of these organisms are excellent models to understand whole body context of behaviors, and we really need to look at whole bodies to understand motor control and reafference. And we need to understand the brain. We also need to understand the body. And the small animals have great potential because we can reconstruct them entirely by electron microscopy and study them in large numbers. And many systems have now genetic access. And there's a lot of very interesting biology to study. And the final notes to computer science. So this is a video showing how the Tesla machine learning algorithm was completely fooled by, and it thought it's an endless trail of traffic lights. See, it detects traffic light after traffic light, doesn't understand what the hell is going on. It turns out that the truck in front of it was carrying these traffic lights, but the Tesla didn't get it, completely didn't get it. Now, why is that? Because probably it didn't understand the reference of motion. I mean, the traffic light moving relative to the ground or the frame of reference, that doesn't really make sense, or only makes sense if that's on a truck. But somehow Tesla's training algorithm never encountered the situation. And of course, if you train the Teslas on just these very simple 2D pictures, where there is no depth information and no movement information, that's going to be completely misleading. And you will not get to a real appreciation of 3D relationships. So what I would suggest to Elon Musk is to read the book from James Gibson, and actually for every computer scientist who is interested in, for example, computer vision, this is absolutely an essential read, because this explains exactly this reafference principle in vision. So how the movement of the body or the eye movement is essential to interpret the visual scenery. And of course, this is, the larger take-home message is that we cannot imagine as computer scientists or biologists the brain as this problem-solving machine floating in this magic solution, like this mother machine, and then spitting out the answers. You need to understand this, or also train the models in the framework of real bodies and real motions and real 3D phenomena. So I would like to stop here and thank my group and our funders, and I'm happy to take questions.
Thank you. Very, very interesting and provoking. Any questions? Oh, I have one. Like you were showing, you are focusing on the importance of understanding the whole body in order to understand the organism, and everything is connected. But is it possible also to understand how they interact with other organisms? Because when we were showing those videos, and you kind of know the models that represent each organism individually, but how they interact between each other? Do they change the behavior because in some other nature? Because I was thinking about, even before you showed the Tesla, like if we could use these models and how those tiny organisms interact to help us understand and to model like the interaction among autonomous vehicles.
Yeah, that's an interesting point. I think this is the area of like collective animal behavior, and these larvae, so far we haven't seen any indication that they behave collectively, not like, I don't know, ants, or it's also known that Drosophila, they can behave collectively. And I think that's a very active research topic that could be highly relevant for robotics and so on.
I could have a question.
Hello, can you hear me?
Yeah.
Thanks for the presentation. It was quite interesting. Yeah, and I think there are many opportunities here for computer scientists. So I have in fact many questions because I found really cool the presentation. First one is similar to what Diogo was talking about, but more about this, that you said that you needed to have this whole body approach, right? So you cannot understand the brain without understanding the body. So, because in the beginning of the presentation, talk about that model, right? That is a 3D model, right? So how do you show that you have to have that, let's say, complicated model, right? Because maybe you could see that it's just one, a more simple model that you just need a particle and this thing is interacting or doing some movements. But why do you need to have this more complicated model?
So in the model, we tried to capture the natural swimming trajectories. So we started with modeling with this sphere that moves in this helical pathway, and then we started to analyze how light can modulate that by introducing into the model kind of steering rules that come from experiments. So what happens if you illuminate this eye or that eye? Then we could measure that this and this side of the animal slows down. So you can incorporate this into the model. And then with very few assumptions, we get phototaxis in the model. So and then you can start to take away some of the components like rotation or how much it's wobbling and so on, and understand the parameter space under which you get behavior. Mm-hmm. So it was a relatively simple model, I would say.
Okay, so you create a simple model and you start to add kind of features, and then when you start to explain the behavior, so then you're good, basically.
No, we didn't have to tune the model too much. We just said, okay, these are the features, helical swimming with these and these parameters, and steering rules, and this is the directionality of the eyes. If you put these three together into a model, you get, and you provide directional stimulation, you get phototaxis. Mm-hmm. So this is a model that you can play yourself with it. Let me just see if I find it. It can be downloaded as executable and run on your computer. Yeah, I will locate this and I can send it to you.
Cool. Yeah. So related to that, because I really have many questions because I found really cool. So the second, so I was thinking, if now that you have a model, if it's possible to try to understand the evolutionary mechanism that led to that animal that won, how evolution how this organism evolve into this new one. Because if you have a model, maybe you could start to think a way to model, the whole evolutionary phenomenon.
Yeah, this is a great point, and this is something I discussed in a paper about the evolution of Phototaxis. Not particular for this organism, but for single-celled organisms. There is also there are also many examples of Phototaxis. So I'm just putting this in the chat. This paper, you can have a look, and there at least you can build up a model of evolution of at least 4 steps of complexity. And the easiest is that you have an organism that has a light sensor, and whenever light comes on, it stops moving, because it like shuts down its cilia. And this could already be important environment. For example, a shadow response or light shock response. There are several protists or single-cell eukaryotes that do this. And then you have to, if you start adding some kind of shading mechanism, it could be sometimes it's a chloroplast that is modified, in like algal cells, or really some kind of accumulation of some pigment. Then you will get, because most eukaryotes swim in this helix, so this is already given, because this is the most natural way to swim. If you just make an object, imagine of a random shape, and you put some kind of a motor on it, like a jet motor or a beating cilium, you will then essentially get a helix. It's very difficult actually to get a straight trajectory. So in locomotion evolution, helix is always before the straight trajectories. All bacteria move in a helical pattern. So that's basically given. And this gives you this scanning mechanism already. Once you have pigment, then you get the periodic shading, because light usually comes from the water, it comes from the top. So you will get this periodic signal. And if this periodic signal can be coupled to any kind of deviation of the movement, like you just wiggle your cilium a bit, you automatically get orientation, because you do the wiggling until you are completely aligned with the light. So it's actually quite interesting, and you could build really a computer model that evolves Phototaxis in 3-4 evolutionary steps. It would be a nice project, yeah.
I have one question regarding the networks you were showing. I think you said that, I don't remember exactly the number, but many different types of cells, and then you show the communities with different colors. I was wondering how those models or those networks are built. Like it's from computer vision, so they identify the nodes, and they also are able to distinguish between which type of cells are different parts. I'm wondering, I think the question is, who determines the classification or the communities? Is it computer vision, or is this a network community detection that detects based on the levels of interaction?
Yeah, it's network community detection. So like Leiden algorithm, this well-known community detection methods that we used. Yeah, and to build a network, that's from the electron microscopic images. So you go through and trace the cells and reconstruct the morphologies, and then find the synapses and connect them into a network. That's how we get the network from EM reconstruction. And once you have the network, you can play with it with all the iGraph or whatever packages. Basically, that's from then on, it's network science.
I imagine that there is no way how to validate these networks, because is there, you can see, right? You trust the algorithm is reading correct and make the connection, and from that you build the knowledge. But yeah, there is no intermediate step, I imagine.
You could validate. One way to validate is to check the left and right side of the body. That should be very similar. I mean, most bilaterian animals are very similar, with some exceptions. The nervous system is quite symmetrical. And this we checked, and there's a very good correlation between the sides. You can, of course, look at another specimen, another individual, and see if you get the same network. This we have done to some extent to one part of the brain, and we find a very similar network. So there are ways to validate, but it's still very time consuming, and it's many years of work to get one such network. But it's improving. The throughput is really improving.
Yeah, no, it's impressive. Like I would imagine that you have like regions with communities, but no, the communities are actually overlaid throughout the entire body, basically, right?
Yes, yeah, many of these communities span the entire body, like this community that controls the cilia, that includes these giant neurons I showed that span the body. That's a community that is responsible for all the cilia in the body. So it's not just one small part of the brain, but really it's a network that is spanning the whole body, and many of the networks are like that. And again, these are really the functional modules of the nervous system. And that's why I say that also understanding really the entire wiring of the brain, you need to look at the whole body. And this is a simple animal, you could say. So imagine the mouse. So I'm sure it's going to be the same, no? Because a motor signal comes from here and you move the tip of your toe on the other end. So that's one kind of behavior that will require the entire body spanning mechanism.
Great. Any other last questions, because we are running out of time?
Yeah,
Yeah, please, go back.
thanks. I guess you can't see me because my camera doesn't work right now, but
I can hear you.
Yeah, go on.
So my question was about the recording that you showed about the cilia, and it seems like the cilia were always moving, and there were just some short period of time when they stopped. Is that right? So they are always active, and the movements are basically made by some short period of stopping.
Yes, so that's interesting actually, that it's not like the muscle that you need a neuronal signal to contract the muscle. The cilia is the opposite. No? They just beat by themselves. And when you give the neuronal signal, that's when they stop beating. And actually it's an active, we could show that those ciliated cells have an active spiking. So they make action potentials as long as the cilia are closed. They have to keep them closed. Don't move, don't move, don't move. And when it stops, then we can move again. So like the default state is movement, and you actively need to stop them. That's what you mean?
Yeah, that's what I was wondering about. It seems a little bit doesn't it like waste a little bit of energy? Because
yeah. It seems it's actually probably more energy to keep them not moving than moving. Yeah, it would be interesting to calculate. But these cilia are beating, they have thousands of cilia and they beat for many, many days as these larvae swim and grow without any food. So just the yolk kind of they have from the mother, that should have enough energy to maintain the beating of these thousands of cilia for many days. So it's incredibly efficient, actually, motor, the cilium.
Okay, thanks. So I just wanted to make a comment about there was this computational neuroscientist called David Marr. I don't know if you've heard of him. He had an interest in vision as well, and basically, I mean, this system will be a dream come true for him. Unfortunately, he's dead, but he what he said was that in order to understand, something like the brain, there are basically 3 levels of investigation, and you have to go from the bigger level to the smaller level. And so the bigger level is basically, what does the brain do? And here you already know the answer. And then the second level is, what is the algorithm? Okay, what is the way that the brain can do this? And the third level is, how is it physically implemented? And he said that in order to get to this question, how is it physically implemented, and basically record neurons and their network and figure out what the neurons are doing and the network mechanism, you need to know what's happening in the first and second level. And currently, we cannot do that with most of the neuroscience systems that we are looking at, in the invertebrates. We don't really know what the brain is doing, and we really have no clue about the algorithms, but we keep recording from neurons and trying to find some mechanism. And basically what you were saying is that this is like trying to understand how birds are flying by looking at the anatomy of their feathers. And of course, that doesn't tell you anything about aerodynamics. So so maybe your system can tell us something about the aerodynamics of the brain.
Yeah, very thanks for the comment. Yeah, I agree that there is a great potential for these kinds of systems, and currently neuroscience is to a large extent mouse focused. So for obvious reasons related to health and so on. But really these fundamental principles of motion and embodied cognition and reference, I think we could learn a lot from simpler models where you can also look at not just the feathers, but everything.
Thank you.
Thank you, Gaspar. I think that's it. We are over time. Thank you so much for accepting this invite and talking to us this very nice presentation.
Thank you very much for the invitation.
And thank you everyone else for being here.
Thanks.
Bye-bye.